evening, everyone. I am Shailesh, and on behalf of Haystack Analytics, I welcome you all to today's webinar series. So today's topic for the webinar is resolving the Klebsiella pneumoniae mobilome, the power of hybrid sequencing in AMR research. So before we start with today's webinar, I'll just like to give you a short introduction about what Haystack Analytics does and a short rundown of our journey. So Haystack Analytics is basically a health tech company based out of IIT Bombay. Our mission is to provide genomics to the forefront of healthcare. So we are making genomics products for tuberculosis genomic solutions, which are supported by BIRAC as well as launched by the Prime Minister of India. Our flagship product, which is infection, aligns with WHO's Sustainable Development Goals and is used for detection of sepsis from patients who have fever of unknown origin. Through genomics, our focus is to fill the gaps in healthcare by creating scalable solutions to benefit patients as well as the entire healthcare ecosystem in India. We are supported by several different partners, including some of the top labs, diagnostic labs in India, as well as government agencies such as the Ministry of Health and Science Technology, BIRAC, CAHO, Nidhi Prayas, etc. We are also partnered with our tech partners like Intel and GE Healthcare for different purposes. Okay. So to give you a brief idea of what Haystack does and what our journey has been so far, the company, since its inception in 2019, has been committed towards working on providing genomic solutions for healthcare industry. So our first product was for detection of TB, which is tuberculosis. So as you would know, Mycobacterium tuberculosis infection is widespread in India, and for detection as well as correct treatment, it is important to know what is the drug response profile of the organism. So our first product, which is the TB whole genome sequencing, it is the first nationwide deployment of TB genomics, wherein we are able to detect not just the Mycobacterium as the organism, any other tubercular microbacteria which might be present, as well as that provides DST, that is drug susceptibility, for 18 antibiotics, right? So that is something that we started off in 2021 as a pan-India program. During the same time, because of the pandemic, we were also involved in COVID genomic service for BMC, where we maintained a real-time dashboard of all the different strains that were coming up in different parts of the city, all through genomics. In 2022, our TB product got the BIRAC Big Grant, which was a big step up for us, because of which we got the funding to deploy the entire program pan-India and to spread across several different centers as well. During 2022, we also started working on our second product, which is now our frontline product, which is Infection NGS. Infection NGS is basically detection of all bacteria, all fungi, using a single test and on premises, all within 24 hours using the power of Oxford Nanopore sequencing combined with our proprietary software. So all of this through 2022 and 2023, we were able to achieve several different milestones across both these products, including infection as well as TB whole genome sequencing. In 2024, we won several different awards, including some national awards by Government of India as well. In 2025, our facility, which is our lab located in Mumbai, got its NABL accreditation for all the products that we provide. And in 2025, during the last quarter of 2025, we were able to win the Tycon East in Boston in life sciences, as winner as life sciences as well as in healthcare, right? So that's about our journey. That takes me to today's speaker, who is Dr. Devarshi Gajjar. She is professor and head of microbiology department at the Maharaja Sayajirao University of Baroda. So ma'am's work also aligns a lot with our work that we do here, which is basically against identifying infectious diseases and antimicrobial resistance. So to tell you a bit about ma'am, Dr. Gajjar has been at the forefront of microbial genomics and antimicrobial resistance research. So she works on whole genome sequencing, multiomics, as well as molecular microbiology. Her core area is pathogen evolution, virulence, and drug resistance, especially focusing on Klebsiella pneumoniae, Salmonella typhi, and few other fungal pathogens as well. Apart from her research work, ma'am, like I said, is also the professor and head of the Department of Microbiology, which is a prestigious institute, as we all know, at the Maharaja Sayajirao University of Baroda. She's also the coordinator for the MSc Biotechnology program, which is also a part of the same department. And ma'am has been leading research in genomic based diagnostics. Of course, she's also funded by several institutions of India, such as DBT, ICMR, DST, and others. So, ma'am's contribution towards genomics, towards infectious disease research is unparalleled. And ma'am, we are truly honored to have you as the guest speaker for today's webinar. Over to you, ma'am.
So thank you very much, Shailesh. It is a pleasure meeting you after such a long time. So I will share my screen.
Yes, ma'am.
You can see it, right? Shall I?
Yes. Ma'am, just try going into a slideshow.
Yeah. I'm stopping my video.
Okay.
Yeah, now?
Perfect, ma'am. We can start.
All right. Okay. So thank you once again, Haystack and Shailesh and team, Chandni, Sakshi. So many people have been interacting with me in the last 15-20 days. It is really a pleasure to interact with all of you and share our work. So today I'll be talking about some of the work, some of the examples and work that we have been doing since the last 10 years. We started with Klebsiella pneumoniae in 2016, and it seems like it's just yesterday that we started understanding this pathogen. So, as Sailesh already told, we have been doing a lot of whole genome sequencing in the last so many years. Many pathogens, different experiences with each pathogen, fungal, bacterial, many bacteria. But one of our favorite is always Klebsiella pneumoniae, and in the next 15 minutes, you will know why we like Klebsiella pneumoniae so much. So, what I'll be doing is I'll be telling you why we try to understand Klebsiella and antibiotic resistance in Klebsiella, how we narrow down. Basically, we are in a state university, so we don't have enough money. We don't have so much money that we can, sequence each and every isolate that we get from the hospital. So, I'll be trying to tell you that how do we choose which isolate to sequence, when to do a short read sequencing, when to do a long read sequencing. I'll also give you an example of two important clonal groups which are converging in India, sequence type 147 and 232. Also, some example about when you have multiple copies of some genes, how does it respond to the phenotype of that isolate? And lastly, a little bit on plasmids, particularly the mobile genetic elements. I am not touching about all the mobile genetic elements. Trust me, there is a lot of data on prophages and insertion elements, transposomes, and a lot of it is there, but I'll just be touching upon what we do with plasmids. So, why Klebsiella? So, let's just consider 50 years ago, there were two different strains of Klebsiella. One on the left, you can see is the hypervirulent, and on the right, you can see is the classical. So we call the hypervirulent strain hypervirulent because it can cause infection in otherwise healthy individuals. It can cause liver abscesses. It can spread throughout the body. It can cause meningitis. And this is mainly because it has a virulence plasmid in it, as shown over here. Whereas the classical Klebsiella pneumoniae was classical because it was in hospital acquired infections and it was particularly susceptible. Then, in the last 20-25 years, what happened is that certain clonal groups mentioned over here, ST11, 147, and 248, somehow acquired the carbapenem resistant plasmid, which is shown in purple color over here. So the classical KP became the carbapenem resistant KP, whereas the hypervirulent KP still was a hypervirulent. And these two strains were completely separate. They were not interacting with each other. They were different. and they were at different locations. Also, the clonal groups, the lineages, everything was totally different. Then one fine day, what happened was that there were some insertion sequences that transcriptionally mutagenized the capsular locus, and these isolates became without a capsule, and because of which the resistant plasmid was, because it was a conjugable plasmid, it came to the hypervirulent Klebsiella pneumoniae. So now what happened? The hypervirulent one became a carbapenem resistant HVKP. So it is CRHVKP. And then because the virulent plasmid was non-conjugable, but it has a helper plasmid now, which is the resistant plasmid. And because of the helper plasmid, now the virulence plasmid has also gone to isolates that were earlier carbapenem resistant. So we have two different clonal groups. One is CRHVKP and another is HVCRKP, depending upon when did a particular plasmid come in a particular isolate. Then in the last 10 years, what has happened is that these resistant plasmids have spread, virulence plasmid have also spread, and because of the high frequencies of homologous recombinations and site-specific recombinations in Klebsiella, this has come across and this has generated into a so-called hybrid plasmid, or which we also call convergence of virulence and resistance. So this, in the last 10 years particularly, mainly reports were from China, but nowadays we are also seeing reports from India that we are seeing hybrid plasmids that were not earlier present 10 years ago. And not only that, the site-specific recombination is also seen between the chromosome and the hybrid plasmid. So now some of the genes that were there on the plasmids are now have gone to the genome level, and so they have now permanently they are residing on the Klebsiella genome. So in other words, Klebsiella pneumoniae, if you read all the reviews, is considered to be an AMR trafficker. Particularly some of the clonal groups that are mentioned in this review. This is a wonderful review. I insist people should read this review. And some of these clonal groups are really spreading. They are super spreaders. People call them super spreaders. And the entire world is currently trying to understand what makes a super spreader a super spreader. Why is it spreading so much? Why is it acquiring plasmids so fast? And once a plasmid comes to Klebsiella pneumoniae, why are these plasmids now seen in Acinetobacter, Pseudomonas, E. coli? And because of this feature, people are considering KP as an AMR trafficker, but there is not so much of recombination and so much of spread seen in the other pathogens. So this is why we want, and the entire world wants to study Klebsiella pneumoniae, left and right, in and out, and what's happening in it. So how do we start our work? We usually collect isolates from hospitals in and around Baroda, let's say Sterling Hospital and Toprani Labs and many other hospitals. And then the first thing we do is that we categorize the isolates into MDR, XDR, and PDR. This is extremely important for us because then we will know how important the bug is to the clinician. And a word from the clinician is extremely mandatory for us, because then we will know that whether the isolate that we have got, whether it is really important or not. And so the first, the first shock of our life we got in 2017-18 was that when we saw that a patient with urinary tract infection died, and which was so unusual, and the clinician was also very worried. And then we found out that that was a pan-drug resistant isolate. It was also resistant to colistin. So that is when we decided that, okay, now it's time, we have to whole genome sequence this isolate. And that's when we started sequencing a handful of organisms. So the first sequencing we did was only for 6 of our isolates, and then the rest is history. Now our lab has more than 500 isolates that we have whole genome sequenced. So this is how we begin. The moment the isolates come to our lab, we first again verify all the resistance parameters with MIC categorization. Sometimes we also do ESBL, MBL assays. Earlier, we used to do multiplexing PCR to identify the genes, but now we know that there are so many genes that it's really practically impossible to do a multiplexing real-time PCR. to find out how many genes are there. So then we decide that based on the importance of the pathogen, based on all these parameters, we decide let's now do whole genome sequencing. So this first piece of data was done by my second PhD student in the department, Siddhi, and she was the one to first sequence just 6 isolates. So we first sequenced 8 isolates with Illumina, and then out of those 8, only one was selected for long read sequencing, Oxford Nanopore. And then once we got this data, it is very easy to fetch all the data from the NCBI and BBBRC and Pathogen Watch, and then we compare our genomes with all the available genomes. So this is not very difficult to do. All we need is a decent laptop with a good RAM, and we can do that. So this is the in general pipeline that we use for our data analysis. We take raw data from the companies. We have tried almost all the available companies in the market, starting from MedGenome, Eurofins, and all these companies, and all are just the same. And we are, let's say, happy with all of them. So after we get this, so now mostly, when it comes to bacteria, people feel that bacterial genome is very small and all you need is just a 0.5 GB data. But that's not true. If you want some decent information from the genome, we generally try to get 1 GB data per isolate paired end. And that is when, if at all, there is any novelty in the genome or any novelty, any variant in the EMR genes, only then we will have some confidence if we have this kind of a data. And only then we will know that which isolate should proceed for a Oxford Nanopore or a long read. Now remember, we are doing this because we don't have money. If we had enough money, we could do hybrid for all our isolates. So very quickly, I'll start with the results. So this is an ITOL image, and as soon as we get our data, the first thing we do is we have to create an ITOL image. An ITOL image gives us information, a snapshot of all the genes that are present in one go. So if you can see over here on the left-hand side, there are MDR isolates, there is one PDR isolate, there is one susceptible isolate, and you can see all the boxes in the susceptible isolates are empty. So the filled boxes means that these are the genes are present, whereas the empty boxes means the genes are absent. So you can nicely correlate that the susceptible ones does not have all these genes, but the resistant and the PDR ones has all these genes. Also, we try to put in all the plasmids. So all the green boxes over here are the plasmid replicons, and all the purple ones over here are the virulence genes. So in one image, we can get an entire information about all the AMR genes, which are in blue, the plasmid information, which are in green, and the virulence information, which is in purple. So one of the most important features that we got in the PDR isolate was the MGRB mutation, which gives us the colistin resistance. And this, at that time, this was in 2017-18, not many people were getting colistin resistance in Klebsiella in India. It was very much reported in China and other places, but in India, people were not getting so much of colistin resistance. So that was something very different we found. This is the susceptible one, and this black box tells you all the genes that are present in the PDR isolate. So this information we got that we have some unusual information of the PDR. Another information was that we found both carbapenomases in the same isolate. We found NDM5 and OXA181, both of them were present. We also found the fluoroquinolone mutations. We also found the MGRB disruption, and this was enough for us to decide that, okay, now come on, let us send this isolate for an Oxford Nanopore sequencing. So once we sent the isolate for long read sequencing, another unusual thing we found was that the NDM gene that we found was on the chromosome. Again, that was not earlier reported. So these are the 4 isolates where I'm showing the data, and you can see that the NDM5 is on the chromosome, which is again extremely unusual, and till date, very, very few isolates across the world, they have shown that the NDM is on the chromosome cluster. So this is the data of PlaCNet, where you can see that the NDM5 is in the chromosome, whereas all these other faraway dots belong to the plasmids. So then again, further surprises with our long read data that it was NDM was present in two copies. So you can see one copy is on ink F2 plasmid, whereas the other copy is on the chromosome. Not only that, OXA81 was both the copies were present on the chromosome. So the short read data was not very confident, and it was not able to tell us whether it is on the plasmid or on the chromosome. Again, it was a few years ago. Now the tools are more refined, and now even the short read data gives us a little more confidence than which we used to get in those days. And the third surprise we got was that the CTXM was in three copies. Two of the copies were on the chromosome, whereas one copy was on the plasmid. So this information that we got using Oxford Nanopore was not found with the short read data. Again, we found all these insertion sequences and transposons that were not earlier reported, that we were able to report in our isolates. So this was something very unique, that NDM was on the chromosome, and there are multiple copies of carbapenemase and ESBLs in this one isolate that we got. Another NGRB disruption that we found was of an insertion sequence 5, which was originally reported in E. coli, and it caused the bypassed charge distribution on the cell wall, and that is how colistin resistance was found in this isolate. So we showed that this insertion sequence was earlier reported in E. coli and a few other organisms, but it was not reported in Klebsiella, and we were the first one to report it in Klebsiella pneumoniae. So nowadays, you can see there is a huge, repertoire of insertion sequences and plasmids. There is association of insertion sequences along with plasmid replicons. So this is an interesting paper. This is not our work, but in this paper, the authors have shown which insertion sequence is associated with which kind of plasmid replicon, and people are trying to figure out what this means. And recently in 2026, there was a Nature paper, and again, Shailesh, if you have the time, please read it. Here they have knocked down, or they have deleted the insertion sequences from the plasmid. So the three panels over here you see, the lowest one is without the OXA48 plasmid, without the insertion sequence on the plasmid. The middle one is the plasmid with the insertion sequence, and the top one is an double copy of an insertion sequence. And you can see the mutations they have reported over here, and it is very well now accepted, and that in presence of insertion sequences on plasmids, the rate at which the disruptions and the rate at which gene inactivation happens is far, far more than in absence of insertion sequences. So you can read the title of this paper, which says plasmids promote antibiotic resistance through insertion sequence mediated gene inactivation. And this is really a very good paper with lots of genomics with wet lab, and it is saying one of the important aspects of mobile genetic elements and insertion sequences. So in the same isolate, we also got RAMR, which is a transcriptional activator of efflux pump, and we saw that it was mutated, and that is what gave tigecycline resistance. Again, in Klebsiella, tigecycline resistance was not reported back then. Nowadays, a lot of people are getting. So this is what we concluded, that coexistence of carbapenemase genes, mutations in OMPK5 purine, along with NGRB disruption gives rise to a PDR pan-drug resistant strain of Klebsiella pneumoniae. We were also able to characterize all the other plasmids in the genome, all the PARG plasmids, all the integrative conjugative elements. There are big islands on the Klebsiella genome which are called ICEKPs, and they are also conjugable. So a piece of DNA can just come out of the chromosome and go to another region. So all of this is very much unique to Klebsiella, which is not found in many other bacterium. So in other words, I just want to tell you that it is because of long read sequencing we were able to characterize all these different things, which was not earlier possible with short read. Then what we did was that we collected all the genomes available on the BBBRC, 217 of the clonal group 147. I'm sorry, this is a very busy slide, but I have simplified it over here, and you can see we were able to see the differences in the carbaphenamase genes with respect to continents. The color coding is given very nicely over here, that in Asia it's always NDM and OXA48, whereas in Europe and America it's always KPC, WIM, IMP, and all these other carbaphenamases. So if someone is trying to make an RT-PCR kit or a lateral flow assay, then selection of a gene is very important because it is next to impossible to create kits for so many genes, and therefore I think whole genome sequencing is the best thing to do, which Haystack is doing, I believe. So then I will move to the next story, which is about clonal group ST232, and this is again a busy slide over here, but I have split this, and I want to tell you that this particular clonal group ST232 is only mainly found in India and Thailand. Again, we found dual carbaphenamase producers over here, and we did not find NDM. So if the clonal group is 147, then it has NDM. But if it is ST or clonal group 232, then it has CTXM and OXA, and it is a dual producer. It is not a single producer. All the strains have fluoroquinolone resistance, 100% carriage of QRDR mutations. There is co-occurrence of two different kinds of plasmids, CoLKP and CoLKP3, as mentioned over here. And surprisingly, the virulence plasmids were not found, but the virulence genes were found on resistant plasmids. So this shows that there is convergence, and 74% of these isolates are converged in India and Thailand. So this is another story of ST232, where we believe that extensive misuse of fluoroquinolones and carbaphenams in the past few years have contributed to a selective pressure, and therefore both of these lineages, ST147 and 231, are high risk clones in India, and they are actually on the verge of becoming pan-drug resistant. And if a person is continuously giving stresses to these strains, then they can shift to pan-drug resistant any minute. And convergence is observed in ST231 clones of South and Southeast Asia, and that is a dual risk. Now, because we were finding too many isolates with dual carbaphenamases, we thought let us find the MIC differences in single versus dual carbaphenamases, and you can see over here we have isolates M40, which is susceptible to both the carbaphenams. M52 has only OXA232, and you can see the differences in the MIC value. NDM, the M39 has only one gene, and you can see the MIC. So if an isolate has only ST232 versus only NDM1, then OXA232 is even more worse than having an NDM1 gene. And at the same time, if there are dual producers, so let's say there is a combination of NDM5 and 181, and NDM1 and OXA232, then the one with NDM5 and OXA181 is going to be more, bad compared to the rest of the combinations. So there is a lot of data in this paper, but I am not sharing all that data. I just wanted to emphasize on the genotype to phenotype association and what happens when there is a single carbaphenamase versus when there is a dual carbaphenamase. And not only that, the kind of carbaphenamase also matters. Now, the MIC value for attributing a isolate to be resistant is just 4 microgram per ml. So these values are way beyond the in vitro lab values are way beyond the 4 microgram, which is clinical value, which is shown. Now I shift to the last part of the talk, which is about plasmid comparison. So here, on the left side of the slide, this is the data of short read, and you can see when we use some of these tools like MobSuit and all these tools, you can see that gave us that this isolate PK17 has 10 plasmids, and you can also see which plasmid is associated with which gene. And on the right side, when we did a nanopore, an Oxford nanopore, that data gave us that this isolate only has 5 plasmids. Now, why is this happening? Because there are some plasmids that have, so the plasmid typing is done using its replicon type. So there is a tool which just detects what kind of replicon type is there. And when you have short fragments of short read sequences, each replicon type is considered as a different plasmid. But when you do a long read sequencing, you can now tell that uh, there are multiple replicon types present on one particular plasmid. Not only that, in this case, again, the short read sequencing missed some of the genes. For example, TEM is present on two plasmids. As shown over here, it is present on INKF2 also, and it is present on this plasmid also. You can also see how many of these plasmids are conjugative and so on and so forth. So what I'm trying to tell you over here is that because we are trying to do some kind of fundamental basic research and trying to understand the antibiotic resistance of a particular isolate, we are really very, very myopic, and we would like to know everything about each and every isolate that we have. And uh, right now, what we are trying to do is go beyond the simple tools that are there and try to go even go deeper and try to understand the promoters and try to understand the expression status of when the gene is on a plasmid or when it is on a chromosome and so on and so forth. So there are many other labs doing the same, but yeah, we are also trying to just do a little bit from whatever we can. So I have many, many such plasmic maps for so many of my cultures over here, but I'm not going to bore you with all these maps. But I'm just trying to tell you that only if you have a hybrid sequencing or a long read, Illumina plus long read sequencing, then only you can have confidence in some novelty. So, in short, what I would like to say is that because we are a fundamental lab and we want to understand AMR, and that's why for us it is extremely important to ask the right questions before we proceed for short read or long read. And what we do is that if we get some variation or something unusual in a pathogen, or we find something that the clinician tells us that okay, this is not being, it is showing susceptible on the plate, but it is not, the patient is not being treated, only then we then consider it for sequencing. So with that, I am extremely grateful to have wonderful students in my lab, and most of the data that I showed today is of Dr. Siddhi Desai, Dr. Suraj Shukla, and Haley, who is presently winding up her thesis. And of course, all of this would not have been possible without my wonderful, wonderful clinical collaborators, Dr. Jyoti Patankar in Sterling Hospital. So she was the one who gave me a call when the patient died, and Giti Maheshwari also has been really helpful in, telling us that just collect this isolate and no, this is not important and so on and so forth. Because we don't have so much money to sequence each and every isolate that we get in the lab. So currently, we are a part of the global CLEBNET GSP epidemiology consortium, where there are more than 50,000 genomes worldwide that are being analyzed. And we have been, like, along with us, a lot of people from many, many countries are taking place. And Dr. Kelly Byers and Kat Holt from Monash University, Australia, are the group leaders for this consortium, epidemiology consortium. I will always be thankful to Pushpendra because it was because of him the genomics work started in the lab. And Ratika, of course, she's an immunologist, and most of our immunology related projects that we do in the lab, we are also trying to understand what difference does the capsule make and how the capsule affects the phagocytosis. We do a lot of blood work and cell culture work in the lab also, and that is all thanks to Ratika. And that's it. And thank you. If at all you want to visit Baroda, you're most welcome, and I'll be happy to meet you. So I think that's all. Hello. Yeah.
Yes.
Did I finish in time, Sailesh?
Yes, ma'am. Yes, right on time.
Yeah. Okay.
Thank you so much, ma'am, for the wonderful presentation. I think we all have learned a lot more about Klebsiella pneumoniae today.